Hello everyone, I am Cécile Herat and I am a biology researcher at the French Alternative Energy and Atomic Energy Commission in a cytogenetic laboratory. In this lab, we are experts in biodosimetry and we develop different cytogenetic techniques that allow retrospective dose assessment at individual level. More particularly, in this tutorial, I will explain you the method of telomere and centromere fish staining, a technique that facilitates and improves the analysis of the three classic cytogenetic techniques that we are daily performing. I will tell you more soon. After the chromosome preparation, using any of the different methods and the spreading on slide, DNA is to be stained in order to be further visualized and captured with a fluorescent microscope that allows then the analysis and the scoring of chromosomal aberration. Basically, DP allows the staining of the whole DNA and chromosome. However, other specific sequences can also be recognized and therefore facilitate the analysis. Precisely, I will describe the protocol that allows the staining of the centromere and telomere using fish probes that recognize and hybridize with these two specific sequences. This staining can be performed on chromosomes obtained by the classic method of metaphase preparation for DCA assay, but also on premature chromosomes obtained with the PCC assay, as well as for the classification of micronuclei. In the figure, you can see the result obtained after fish staining on metaphasic chromosomes. In green, in the center of the chromosome, you can visualize the centromere, while in red, in each chromosome strand extremity, you can visualize the telomeres. If you look carefully, you will be able to see a dicentric chromosome with two centromeres, a ring, and two acentric fragments without any centromere. Here, I show you the different cytogenetic methods used in biodosimetry and allowing the assessment of the individual received dose. The dicentric chromosome assay, or DCA, allows, after GESA staining, the scoring of dicentric chromosome and rings. The staining of telomere and centromere does not change the strategy of scoring, but facilitates the analysis and improves the detection of chromosomal aberration. The premature chromosome condensation assay, or PCC, allows basically the scoring of chromosomic fragments and the detection of excess fragments. In this case, the fish stainings allow the specific scoring of dicentric chromosomes, rings, and acentric fragments. So the analysis is a bit longer, but much more informative. The cytokinesis block micronucleus assay, or CBMN, allows the scoring of micronuclei when cells are blocked in cytokinesis. The staining of centromere and telomere enables micronuclei classification in fourth category. Even if this method required a longer time for analysis, it could improve some parameters of the analysis by providing additional information. I give you an example of the two types of staining for each of the three techniques. Today, we will precisely see the protocol for this particular fish staining. I will provide you with the maximum of details and the trick that we have developed. First, here is the list of the materials you need for the staining, the laboratory equipment as well as the reagents. Work under the chemical rule when it is indicated, as some of the products are toxic. Here is also the list and the composition of the different solutions that you will need to prepare. The day before the staining, you can already throw the slide or take them out of the refrigerator and let them on the bench. Before starting, prepare the solution as indicated on these slides. First, the slides need to be fixed in formaldehyde and prepared for the staining. After PBS rehydratation, the slides are incubated for exactly 2 minutes in formaldehyde and then rinsed in PBS. Remember that formaldehyde is very toxic and that you need to work under a chemical hood. In the protocol, we are using porcelain boxes for all the incubation and you are moving each slide from one box to another. After 3 rinses in PBS, the following step is the pepsin digestion that will clean up the protein from the DNA and make the latter accessible to DNA probes. Pepsin has been prepared and heated at 37 degrees in advance. We move the slides from PBS to pepsin for 5-minute digestion at 37 degrees. If you are staining PCC or micronuclei, incubate only for 3 minutes. It will be enough and better since the DNA is less compacted. Be precise with the time of incubation. Then, quickly rinse the slides in PBS, fix again in formaldehyde for 2 minutes, and continue with 3 PBS rinses. After that, you can start the desidratation with successive incubation in ethanol 50%, 70%, and 100%. After the last ethanol bath, we remove the slides and let them dry for at least 30 minutes. Once dried, the slide will be ready for the DNA probes hybridization. You can anticipate by switching on the hot plate, preparing the PNA probe solution as indicated in the list of solutions, and humidifying the hybridization box. Hybridization boxes, as depicted in this slide, are just plastic boxes wrapped in aluminum folder to avoid light penetration. We have placed 2 paper strings on 2 sides of them that we humidify with water before use. The staining consists of the hybridization of PNA probes specific for the centromere sequences and for the telomere sequences. Each probe is coupled with a fluorophore for further detection. We drop carefully 60 microliters of the probe solution on the slide and try to distribute it on the whole length. Then, we protect with a plastic film tailored from a freezer film for the hybridization incubation. Try, if possible, to avoid entrapping bubbles. Then, place the slide on the hot plate set at 80 degrees for exactly 3 minutes. This step represents the beginning of the hybridization. Here, you can see better what we use as hybridization box. Any dark and humidified box can do the job. Exactly after 3 minutes, we remove the slide from the hot plate and let them cool down for a few seconds before laying them for incubation in the box at room temperature. At this point, we wait between 1 and 2 hours for the hybridization to occur, but no more than 2 hours in order to avoid too much background. During the time of incubation, you can warm up the washing solution too in the water bath. Once the hybridization is completed, we remove the plastic film on the slides and wash them twice in the washing solution 1 for 15 minutes, and then twice in the washing solution 2 at 37 degrees for 5 minutes. What remains now is to do the DP staining. We incubate 5 minutes in PBS and then 5 more minutes in DAPI solution before a last rinse in PBS. Now I show you the slide mounting. We take the slide out and dry the back. We drop PPD on the slide with a Pasteur pipette. Then we carefully place the cover slip as you can see in the video. To destruct bubbles, We finally press the slide between paper towels. The slides are now ready to be analyzed. In the meantime, they can be stored either at 4 degrees for a few weeks or at -20 degrees for years in specific plastic storage boxes. For the acquisition, you need a microscope with the 3 following channels: DAPI, FITS, and spectrum orange. In our lab, we have the Metafair acquisition software, but is not necessary for performing the analysis. First, we are making a gallery. The software will automatically capture the cell of interest: metaphases, PCC, or micronuclei. After cleaning the slide with ethanol, the gallery is established with the 10x magnification. We program the microscope to acquire metaphases, and then it captures all of them in the predefined area. After the triage of the gallery, we can start the final acquisition with stained centromeres and telomeres. The microscope will acquire successively the different channels to obtain the final image. Once you have acquired all the images, you can export them for later analysis. In the lab, we are using ImageJ equipped with the HomeMap plugin that we developed. First, be aware that you will not be able to analyze all the images, since sometimes the chromosomes are overlapping too much, and we prefer to exclude images like the upper one. After selection of the image, you can observe a semi-automatic analysis with our CA Detector ImageJ plugin. It is able to delimitate each chromosome and fragments, and then detect the centromeres and telomeres. From there, it automatically detects dicentric and ring chromosomes as well as acentric fragments. You see on the metaphases the dicentric chromosomes circled in red and the resulting acentric fragment marked in green. Of course, you need to check and correct the automatic analysis, but it really saves time. I can give you more details. When we analyze the metaphases, first we check if the cells was in first or second generation, and then the number of centromeres. For metaphases analysis, we consider only cells in first generation with 46 centromeres. For PCC, we accept cells with 43 to 47 centromeres. Then, we validate for dicentric and ring chromosome detection, as well as the different acentric fragments with telomeres at each extremity or only one. We are also able to detect interstitial deletion resulting from 2 double-strand breaks. We can then score either dicentric plus ring or total double-strand break for those estimates.
This staining method seems to be much more informative than the classic Giemsa staining that I am using. But it also requires much more time, and so it delays the acquisition of the final results. Are you sure that in case of an emergency, it can still be used?
Of course, the telomere and centromere staining takes more than the 30 minutes needed for a Giemsa staining. I think that I usually need around 5 hours, but I am sure that you can reduce the duration of the probe hybridization if you really need to save time. Moreover, the results obtained are more robust and the further analysis is really more user-friendly. You don't need to be a specialist to perform the analysis contrary to the Giemsa analysis.
First question about the technique. Why are you using PNA probes instead of simple DNA for hybridization?
You can do it with DNA props also, and how it was made before, but the background noise was important. PNA props represent an improvement of the technique. These props create more stable interaction with DNA and are also more specific. PNA props are protein chains with a glycine backbone and purine and pyrimidine bases linked to backbone by methylene bridges. This structure also contains no charged phosphate group, making the binding between PNA and DNA stronger than between DNA and DNA. Moreover, PNA probes are more resistant to enzyme degradation.
Why are you so sure that the analysis will be easier and more accurate using the TC staining?
I have shown you some examples during the video. The centromere staining makes the detection of dicentric chromosome much more evident and robust. The identification of rings and the different acentric fragments is also much more simple. Just look at this chromosome from stained metaphases. When you use Giemsa staining, you cannot detect dicentric with closed centromere like for the two first chromosomes. While here, with the telomere and centromere staining, it's just evident that they are dicentric. But the opposite is also possible. Without the staining, you can mistake and count a dicentric where it is only a chromosome with a crossing of chromatin. The two last chromosome images let you think that they are dicentric, while it is just two overlapping chromosomes. For the recognition of acentric fragments and rings, the telomere and centromere staining is also very useful. I try to illustrate with the analysis of these metaphases. Look at the ring in number 1 and the acentric fragments in number 2. They are hardly distinguishable without centromere staining, and you can hardly determine in which category they belong. The same for the two acentric fragments in number 3 and number 4. The number 3 fragment contains 4 telomeres and so results from 2 double-strand breaks, while the number 4 fragment has only 2 telomeres and comes from a terminal deletion. The telomere and centromere staining just makes the analysis more informative with the scoring of all unstable chromosome aberration and most likely provides more reliable dose estimates. The telomere and centromere staining is also very informative on PCC and is the only method that allows the detection of the dicentric chromosome. Circled in red, you can visualize the dicentric chromosome, while in green you have the rings and in blue the acentric fragments. Instead of only counting excess fragments, the telomere and centromere staining allows the scoring of the unstable aberration also in PCC.
I see much better now. Do you plan to share your ImageJ plugin, or do we need to develop our own in order to facilitate the analysis?
We are thinking to make the CA detector plugin available only for free use. We are still trying to figure out how to make it possible without having to download the plugin.
I do hope you will make it. Let me know where it will be available. Let's go back to the protocol. It seems simple. A first step of fixation, pepsin digestion, another fixation, drying and staining. The critical step for me should be the microscope acquisition. I'm not sure whether I'm able to make correctly the settings on my own.
I can understand. I didn't describe this part too much, because we are using the Metaverse software from Metasystem, but it is not necessarily required. I will try to provide you with the main step, and you should try to get in touch with the microscope facility of your institute to figure out how to perform the acquisition together. If you didn't do it before, you need to create a gallery. You only need a 10x magnification as a channel for the DAPI acquisition. Either you have in your software an automatic capture of metaphases, PCC or micronuclei, or you can set it by yourself by saving the coordinates of each cell of interest. In case of automatic detection, you need to make the triage of the gallery. At this point, you are ready for the final capture using DAPI, FIS and SPO channel with a 63x objective. I cannot tell you more regarding the time of integration, as it really depends on your specific microscope.
Okay, it sounds clear. You have shown mainly metaphase staining, but you have said that PCC and micronuclei also can be analyzed with this method. Can you tell me more about that?
For PCC, it's quite the same principle as for metaphases. You just need to distinguish human from modern chromosome. For micronuclear analysis, you can visualize directly on the microscope or whatever software you are using to observe images. You just need to score the distribution of the 4 different classes of micronuclei according to the presence of centromere and telomere. You will find micronuclei without any centromere and telomere, micronuclei with only centromere, but they are really rare, micronuclei with the telomere only, that are containing only acentric fragment, and micronuclei with both centromere and telomere containing entire chromosome. This method is still under development and should improve the sensitivity of the micronuclei assay by allowing us to better characterize the micronuclei background levels.
You have just said that the telomere and centromere staining improves the sensitivity of the micronucleus assay. Is it the same for the DCA and the PCC assay?
Yes, we have shown that staining on metaphases allows the detection of up to 30% more dicentric chromosomes, while the sensitivity of the test is considered to be between 100 and 200 mg. With this technique, we can consider a detection limit as low as 100 mg and maybe even lower. The sensitivity of the PCC is quite debatable, but it is generally considered to be between 50 and 200 mg. We can increase the sensitivity with our staining because we are scoring dicentric and acentric fragments and not only excess of chromosome fragments. However, we didn't quantify yet the improvement.
I have another question. To begin with the telomere and centromere staining, can I apply this method in complement to my favorite GIMSA staining?
Sure, you can make several staining on the same slide and reacquire the different staining using the same gallery to compare and complete the different analyses. To destain the previous staining, first make a prolonged incubation in PBS to remove the cover slip and then incubate for 30 minutes in PBS chill 0.5% at 37 degrees.
I have a last question. You are describing the technique for the staining of human chromosomes, but I also work with mice. Can I apply the same protocol?
The telomere sequences are common to all vertebrates, but it's not the case for the centromere sequences that are specific to humans. Maybe you can try to find a specific plot for your species.